Welcome to Module 4, Reprogramming to iPS cells. In this module, you will learn about induced pluripotent stem cells and how they are made. Now that you have learned about adult stem cells and how they are differentiated, it is important to understand that differentiation is not an irreversible commitment. Cells can be reprogrammed. This means that a cell can become pluripotent again. While this does not occur naturally, scientists and engineers have developed several methods that make this possible. Reprogramming previously differentiated cells can be done by several methods, like nuclear transfer, cell fusion, or transcription factor transduction. We are going to focus on transcription factor transduction because of its many benefits. Prior to a study done by the Yamanaka lab, little was known about the factors that induced the reprogramming in these cells. Their lab was able to demonstrate induction of pluripotent stem cells by introducing 4 factors: OCK3/4, SOC2, CMYC, and KLF4. Being able to reprogram in an already differentiated cell is useful because it means that you do not have to use embryonic stem cells to attain a pluripotent cell. Cells of many types can be taken from a patient and induced to become pluripotent, which is a non-controversial and easily accessible way to make a pluripotent stem cell culture specific to a patient. While adult stem cells are also non-controversial and patient-specific, adult stem cell niches are often in difficult-to-reach places in the body. First, let's review what pluripotency means. As you learned in Module 1, pluripotency means that a cell can turn into many different types of cells that can be found all around the body. Second, it is important to understand what a transcription factor is. A transcription factor is a protein that binds to the DNA to regulate gene expression and transcription of the DNA. They come in all shapes and sizes and have a wide variety of functions. When they recognize that their specific sequence, they bind to the DNA and begin transcription. In a single organism, every cell has the same DNA, but based on what type of cell it is, different regions of the DNA are accessible so that these transcription factors can make the correct kind of protein needed for that particular cell. They can be turned on and off based on the different biological processes a cell type must perform. We are going to focus on the 4 different transcription factors we have talked about before, which have the biggest impact on the cell reprogramming. The first 2, OCK3/4 and SOC2, have the function to maintain pluripotency in embryonic stem cells. The second 2, CMYC and KLF4, their function is to modify chromatin structure so that OCK3/4 and SOC2 can bind to their large target regions of the DNA. The CMYC protein also has many downstream targets that enhance proliferation and transformation, all of which we help reprogram the cell. This study was done in vitro, meaning with biological materials, but outside a biological body. Yamanaka inserted these 4 factors into mouse skin cells. The chromosomes unwinded, and these factors could attach to the DNA, overwhelming the competing message from the skin genes. These factors fooled the cell into thinking it's an embryonic environment again, therefore reprogramming it. While iPS cells are a way to return the cells to its pluripotent state, they are not identical to embryonic stem cells. It is also understood that none of these methods mentioned today are 100% effective. While the ability to reprogram different types of cells has been shown thoroughly through many different studies, this technology is still being optimized so that it can be used in a practical medical setting.